Hello, everyone, and welcome back. Today, we are going to be talking about chemical synapses and how uh, one neuron can communicate with another neuron across the synapse. Our learning objectives for today will be to explain how signals are sent from one neuron to another, give some examples of the different categories or classes of neurotransmitters, and explain the ways that signaling is stopped after neurotransmission is complete. So, what we're not going to talk about today is electrical synapses, and electrical synapses are just where the ions flow from one neuron to another through a gap junction. But instead, we're going to be talking about chemical synapses, where the wave of depolarization from the action potential when it gets to the axon terminus is going to cause the release of neurotransmitters or chemicals into the synaptic cleft and be sensed by the postsynaptic neuron. Most synapses are chemical synapses. So a synapse is just a cellular junction, and it is a cellular junction between a presynaptic cell, which is always a neuron, and a postsynaptic cell, which could be a neuron, it could be a glandular cell, it could be the muscle fiber at a neuromuscular junction. So the downstream cell could be one of several different options, but the presynaptic cell is always a neuron. At the synaptic terminus, the wave of depolarization is going to cause the release of a chemical, and those chemicals, those messengers, are called neurotransmitters. In this way, um, neuronal communication is very much like a um, endocrine signal, but it would be a, uh, not endocrine like where the chemical goes through the bloodstream, but instead is is localized to the immediate area, just right across the synapse. So the presynaptic neuron is going to have a biosynthetic enzyme that synthesizes the neurotransmitter, and then another enzyme that is going to transport the neurotransmitter into a synaptic vesicle. Through a very complicated system of proteins, the synaptic vesicles will dock and become primed, and they are ready for a signal. Then, when the wave of depolarization comes down into the axon terminus, a voltage gated calcium channel is going to open. That influx of calcium is going to be sensed by a protein called synaptotagmin, and synaptotagmin causes the T snares and the V snares, you don't need to know what those are, and SNAP25 to twist up like corkscrews, and the synaptic vesicle membrane becomes one with the cell membrane. And so the inside of the synaptic vesicle becomes the outside of the cell, and the neurotransmitters are released into the synapse by exocytosis. And so they travel into the synaptic cleft, they diffuse across by regular diffusion, and they are received on the postsynaptic cell by either ionotropic or metabotropic receptors. So, like I said, the wave of depolarization goes down the axon, and it causes the vesicles to release their contents into the synaptic cleft. We usually think of neurons as being this dendrites and a cell body, and then an axon going off from that with synaptic termini. That's the motor neuron. That's what we usually draw it as. But neurons can take on lots of different morphologies depending on their role. But the mechanism for exocytosis is always the same. So the steps of exocytosis are the wave of depolarization enters into the axon terminus. That depolarization causes the opening of a calcium channel and an influx of extracellular calcium. And that calcium acts as a signal to cause the exocytosis of the contents of the docked and primed synaptic vesicles. Those contents, the neurotransmitters, will then drift across the synaptic cleft and bind to channels. Here I've shown an ionotropic channel. Ionotropic means they are ligand-gated ion channels. So the ligand is the neurotransmitter, and when it binds to the channel opens, increasing the permeability of those ions, and so those ions will flow down their electrochemical gradient. Because, like we talked about with the action potential, because the outside of the cell has much more sodium than the inside of the cell, if the ion channel that opened was a sodium channel, then sodium would go from the outside of the cell to the inside of the cell. And that would cause the membrane potential to go toward the equilibrium potential for sodium, which is about positive 60. So that would cause a depolarization. On the other hand, if the ionic channel was a potassium channel, potassium is much higher on the inside than the outside, and so the potassium would be move down its electrochemical gradient to the outside of the cell, and that would cause a hyperpolarization. So hyperpolarization would be becoming even more negative than the resting membrane potential of whatever it was, negative 70. So it would move toward the equilibrium potential for potassium, which is closer to negative 90 millivolts. So, so depending on what channel it is, what ion is flowing through it, because these channels can be very selective, you're either going to have a depolarization or a hyperpolarization as a result of the neurotransmitter binding to the ion channel. So we call these hyperpolarizations, we call these IPSPs, or inhibitory postsynaptic potentials. We call the depolarizations EPSPs, or excitatory postsynaptic potentials. But either way, you're either going to get closer to or further away from the threshold potential, which was, remember when we talked about the action potential, was when the voltage-gated sodium channels opened. So the direct synaptic transmission involves the binding of the neurotransmitter to a ligand-gated ion channel in the postsynaptic cell. The neurotransmitter binding causes the channel to open and will either cause potassium to flow out of the cell, or chloride to flow into the cell. Either way, that's going to cause a hyperpolarization or an IPSP. The opening of the sodium channel would cause a depolarization or an EPSP. So excitatory postsynaptic potentials are depolarizations bringing the membrane potential closer to threshold and are caused by sodium ions flowing out of or into the cell. Inhibitory postsynaptic potentials, or IPSPs, are hyperpolarizations, and they move the membrane potential further away from threshold, getting less likely to have an action potential. They are caused by either potassium ions flowing into the cell or chloride ions flowing out of the cell. No one EPSP is going to cause a action potential, and so most cell neurons have most postsynaptic neurons have many synapses onto them, either onto their dendrites or onto their cell body, or both. And these synapses can be either inhibitory or excitatory. We can sum them up using one of two things, either temporal summation or spatial summation. Temporal summation would be just if the two EPSPs happened in close time to each other, that might get you to an action potential, to the threshold potential. it usually takes maybe about 10 more EPSPs than IPSPs to actually reach the threshold potential. We could also sum, due to them being close together in space and closer together to closer to that axon hillock, because the threshold potential only matters if it's above threshold at the axon hillock, because that's where the voltage gated sodium channels are. So, so we really got to get to that potential at the axon hillock, and so the closer the EPSPs are to the axon hillock, the more likely you are to have an action potential. And don't forget that the IPSPs have to be added in too, both by spatial and temporal summation. So there are many different kinds of neurotransmitters, acetylcholine, some amino acids like glutamate and GABA, or gamma-aminobutyric acid. So glutamate is the major excitatory neurotransmitter in the brain, and GABA is the major inhibitory neurotransmitter in the brain. It could be biogenic amines, neuropeptides, even some gases like nitric oxide. So, and then in addition, each neurotransmitter may have lots of different types of receptors that could be excitatory or inhibitory. They could be ion channels, like we mentioned, or they could even be G protein coupled receptors, which we call metabotropic, and we're not going to get into that today. So some of the ones that I mentioned, you don't have to memorize all of these structures. I just want you to be aware of the different categories. So one of the categories is acetylcholine, all the neuromuscular junctions, also important in memory and learning. We could have both ligand-gated ion channels and metabotropic, and in the case of acetylcholine, the nicotinic acetylcholine receptors are all excitatory, whereas the muscarinic acetylcholine receptors are all inhibitory. We could have amino acids, like I said, with glutamate and GABA being the two big ones, and also glycine in the peripheral nervous system. So all are known to affect the CNS and the PNS, but the major ones in the CNS are glutamate and GABA. We could have biogenic amines. You probably know some of these, like epinephrine and norepinephrine, or the adrenalins, and some of the important ones for mood and modulation of neurotransmission, like dopamine and serotonin. And we all know, these are important for certain disease conditions. Think of Parkinson's disease as a loss of dopaminergic neurons, and this is substantia nigra in the brain. Serotonin, low levels of that can cause things like mood disorders. One other group is neuropeptides. These are short chains of amino acids that act as neurotransmitters, include things like substance P, which causes you to feel pain, and endorphins, which are going to block the pain sensation. So opiates work on the same receptors as the endorphins, and so can be used as painkillers. At the end of neurotransmission, the way that we end neurotransmission is one of three ways. We could have the neurotransmitter just diffuse out of the synaptic cleft. This is kind of slower and so isn't nearly as active as we would need it to be. So the major ways are that the surrounding cells will take up the neurotransmitter. So we call this reuptake. So there are transporters in the membrane that are transporting, say, serotonin or glutamate or GABA back into the neuron